lipids, what's the conclusion about lipids? Phospholipids, fatty acids, cholesterol, they're all non-polar. Proteins, on the other hand, are polar, they are soluble. And what we want to know about proteins is that everything we're going to talk about for the most part, unless it's a cell membrane or something like that, is going to be about proteins, right? It's going to be all the enzymes we talk about. When we talk about actin and myosin filaments, we talk about a lot of these hormones, right? So these are going to be proteins of some sort or another. As far as the many functions that we're going to see, right? These are just some of the general types from your book, right? Structural proteins of collagen in your bone and connective tissue, which we'll talk about today a little bit. And then all your regulatory functions as hormones, small peptides, your contractile ones, which we'll get into, actin and myosin, which are going to produce movement, right? And, not mentioned here is, all the cell membrane stuff, right? Which we'll get into. But so these proteins are made up of individual amino acids, right? Formed together. They're going to form together to form small peptides, right? But each one of these, right? Carbon with an OH carboxyl group on one side and an amino group on the other side. The important part about these is, they have this polarity to them. They have an amine group on one side, a carboxyl group, but then they have this R right over here, right? This side chain over here, right? And that side chain is really going to confer, because otherwise all these amino acids are the same. The side chain is what confers the properties on each individual amino acid, right? So you can see here's a couple of examples of these side chains right here, right? Just different groups that are going to provide for different sort of properties of each amino acid. And when you string a bunch of them together, they're going to make up the characteristic of the protein. So before we get into proteins though, those amino acids, some of them are directly used as neurotransmitters like glutamate and GABA. These are main excitatory, inhibitory neurotransmitters of the nervous system. And then some of them are modified slightly, like these tyrosine amino acid is made into both dopamine, norepinephrine, epinephrine, right? Undergoes certain modifications, right? Serotonin, big major mood neurotransmitter in your brain, is also another monoamine, right? These basic amino acid derivatives, right? And then you link a couple of these amino acids together, you get these small peptides. Some of the major ones you'll be looking at will be certainly vasopressin or aldosterone, right? Angiotensin. These will be big players in bio 145, right? So all these are protein related or small amino acid related stuff, right? All right, so dehydration and hydrolysis, right? If we want to join two of these amino acids together, right? We dehydrate it, we remove this water molecule, and we form this peptide bond, right? Just like we did with sugar, with the saccharides, right? Except that this is called a peptide bond because it's between a carbon and a nitrogen group right over here. And so proteins will always have this polarity. One end, you'll always call this the N terminal and the C terminal for this amine and CO group over here, right? So this is how you make a protein. You string a whole bunch of these together, formed by these peptide bonds right here. So that stringing together of the peptide bonds, right? The individual amino acids, you form this, what's called the primary structure, right? And that will be written as something like not ATZ, but it'll just be written in a string of letters, right? And, interestingly, just very recently, they finally come up with algorithms and artificial intelligence programs that could be fed this primary structures of amino acid and will ultimately show you exactly what shape that protein is going to make, right? They've been trying to do this for years, but this program, you put it in, it's like 80% or 90% correct when predicting a protein's actual 3D structure over here, just based on the actual string of amino acids, because those are going to interact in stereotypical ways. These hydrogen bonds and other kind of bonds are going to create this secondary and tertiary structures over here that are going to give proteins its shape. But that's all based on this pretty much, right? How they're going to interact with each other and form these complex shapes. And shape is the important point about proteins. Right? So what do proteins do? Well, enzymes are going to do all those synthesis and rehydration reactions, as well as every other reaction is going to be needed to be mediated by enzymes, right? All your mechanical work within a cell, you're transporting stuff from one part of the cell to another, or, the one thing we'll be looking at in depth is the actin and myosin filaments sliding up against each other, right? So this mechanical work, the two proteins working on each other. And then there's also transport work, right? Stuff coming in and out of the cell through its membrane, through these membrane transport proteins. Right? So all these, everything basically is going to be done by your proteins in your body right there, right? So everything we're talking about, almost always talking about proteins, unless we start talking about steroids and stuff like that. And then the other thing about proteins, right? This functional, the structural property of proteins, among other things, when we talk today about connective tissue, this is the dermis, right, of your skin, right? Here's the epithelial layer lining the outside. Underneath is your dermis. Your dermis is largely collagen fibers, collagen protein fibers, or elastic fibers over here. Right? These are major components giving your skin its strength and elasticity, right, by these two types of fibers right here, which we'll talk about in the lab today. All right, so that's another component of proteins, that structural makeup of the body, right? So I guess I'll mention it here. Elastin protein, collagen protein within any tissue, whether you're talking about your dermis or your, your big blood vessels or anything like that, right? With these two proteins embedded within the connective and muscle tissue, right, you have the ability to be really strong, that is resistance against pull, as well as be able to stretch and recoil, right? This is a big factor when we're talking about breathing later, or you guys in 145 are going to be talking about proteins. And that's just examples of when these will come up. And the other big thing for proteins is their membrane proteins, right? All the stuff they're doing for your membrane. In fact, your membrane by itself is very soft and delicate. It wouldn't be able to support all its life-forming functions. So, these two, the proteins and the lipid bilayer, was both necessary parts for life to actually form. They had to kind of form together. They're much more stable when you have these proteins stuck. And then the proteins stuck within them are not only given the structure, but also, the major sort of way that we interact with the environment around it, bring stuff in, let stuff out, right? All these protein channels. And for our purposes, when you're looking at these proteins later on, next unit, you could think of them as blobs and all that. We don't really need to know too much about the protein structure, but they are proteins, right? All these membrane channels, right? But also remember that when we think about that hydrophilic, hydrophobic reactions, right? Different parts of the protein, right? You have those hydrophobic regions embedded around the fatty acid part of the phospholipid bilayer membrane, and the hydrophilic regions facing either the external or internal environment, right? And then maybe this side in the middle here will all be either hydrophilic or hydrophobic, right? But that is getting to that whole amino acid sequence and those side groups that are going to confer these properties on them. The last one, we mentioned glycolipids. There's also glycoproteins embedded within the cell membrane. And again, membrane glycoproteins and glycolipids, one important thing is they're serving as cell identity markers, right? So immune systems kind of recognize different cell types by these glycoproteins and glycolipids. These glycoproteins also kind of provide a slippery surface for moving through, say, the blood, your blood cells moving through the blood. They also form interactions with other cells and water, right? So glycoproteins They're just one another, one of those cell membrane proteins that are having these kind of functions. Next subject, enzymes. Enzymes, right? Enzymes are those catalysts that we talked about when we talked about chemical reactions and what was needed for them to actually get together and have the energy to change or break things apart, break bonds, right? I mentioned that you had to have a lot of them. You had to have a high temperature, maybe they had to get together with enough energy and the proper orientation for a particular reaction to occur. Otherwise, it wasn't happening. Right? And what these enzymes are going to do is make this reaction like 1 million or 1 billion times more likely to happen. And so when we talk about enzymes, right, we mentioned that activation energy, right? Even if it was in exogenic reaction, right? You still needed to overcome this activation energy, right? All that collisions and right orientation, all that stuff, right, is going to be lowered by the addition of this catalyst. And in our case, the catalyst is this enzyme right here. So enzymes, any particular enzyme binds and interacts with specific substrates, like a water and a glucose molecule or a sucrose molecule, to put them together, right? And they catalyze a very specific reaction, right? So we have millions of different enzymes, millions, thousands, whatever you have of different enzymes to carry out very specific functions. And then, as I said, the chances of those things colliding with enough energy and with in the right orientation is very rare. So, you know, this is from the book, 100 million to 10 billion times more rapid, right, than if you had to rely on overcoming this activation energy. And the other point is it's got to kind of put these two molecules together in the right direction, orientation, right? And so this is what your enzymes are doing. Your body, there's all sorts of homeostatic mechanisms, both intracellular and by the body, to control enzyme activities and enzyme like number, right? So how much a particular enzyme gene is expressed and transcribed and made, that's under the control of transcription factors, which are also going to be controlled by other messengers and stuff like that, right? And so there's also within a particular cells, there's going to be like things like coenzymes or enzyme inhibitors that are going to affect the activity of an enzyme, right? So the presence or absence of certain coenzymes or enzyme inhibitors are going to affect how much the enzymes work. And then the cell environment, again, the pH is a big one, right? And temperature and stuff where they're going to become activated or inactive, right? So in your stomach, right, you have enzymes that break down protein, right? Those don't become active until they're in a very acidic environment, which the stomach is, right? So different enzymes have different sort of needs, and then they'll determine how active they are based on the actual chemical environment, right? And then the other just note, if you see the word ACE, right, like sucrase, breaks down the sugar molecule, right? Just kind of get used to looking at these when we say words like ATPase, right? When we're looking at that reaction, those are all enzymes which are catalyzing those particular reactions right here, right? So this will be a big thing, right? Carbonic anhydrase is going to remove the water from that carbonic acid reaction that we talked about, right? That is enzymes. And as far as, the cartoon picture of what they're doing, right? Again, a very particular shape of the enzyme. In this case, they're showing us simplistic kind of little bumps right here that are going to be able to specifically hold a sucrose molecule, right? The sucrase enzyme holds this sucrose molecule, and it's also going to bring water in there. And what it does somehow is like kind of bend the molecule so that it's in a highly unstable state, so that when water comes in, it's able to break the bond, from sucrose into glucose and fructose, right? So the particular shape of the enzyme has a particular place where the substrate fits, and then it catalyzes this reaction right here. And then once it's done, it's like bring on the next one, right? Nothing changes with the enzyme, so it can catalyze many, many, many reactions like very fastly, right? So they'll just be ready for the next one. So this shape over here, the important point when you get along here, maintaining pH homeostasis and all that, if you change the environment in any way or the temperature of your body, right? Your temperature or your pH is going to affect the activity of these enzymes, which could mean life or death, right? So in this graph right here, it's showing the enzyme activity according to temperature, right? So lower temperatures, it's not really working, and higher temperatures is not really working. You have to maintain it. This particular one has a very particular optimal temperature around, say, 37 degrees, which happens to be your body temperature, right? Other than that, it's out of range, and it either works less efficiently or not at all, right? So this whole idea of a protein not working due to pH and temperature causing a loss of shape is called denaturation. Denatured, right? When you try to change the shape because of the environment, right? The big ones are temperature and pH are going to affect that. Right? Here's another example of that. Just the complex shape, you put in pH, it's all strung out, and all of a sudden these molecules, which did fit nice into these substrate active sites over here, are all over the place, and they don't even it doesn't really fit in these spots over here anymore, right? Extreme pH or temperature. So that's proteins, right? The main one, all those membrane transport ones, all the smaller amino acids acting as neurotransmitters and stuff, and then enzymes are the ones that are going to come up quite a bit for proteins. And shape thing, right? Denaturation, remember that word. All right. Last one, nucleotides, right? Most people think of nucleotides about DNA, right? We have those 4 base pairs, right? Thymine, adenine, cysteine, guanine, and they get together to form that double helix by these hydrogen bonds right here. We don't really care about that in physiology so much, right? But while we're here, we can look at the basic structure. Every single one of these has this side groups right over here, the deoxyribose sugar and the phosphate group, right, on each one. And then what's different about them are the bases over here, right? And they're known in this case for information storage. But our interest in physiology will be this one over here, this adenine nucleotide, a little bit with maybe G. You will see GMP, like GMPs and stuff in cellular signaling, but I don't know how much you guys get into that, or CMP, cyclic. But these one over here, this adenine will be big because this is what's going to form the ATP, right? Adenosine triphosphate. So this one over here, that nucleotide structure with this phosphate group, this triphosphate group over here, right, is going to be our main energy currency in our body right over here, right? Because it's carrying these high energy phosphate bonds, which are easily transferable. So when we're, studying this whole metabolism thing, we're bringing in glucose and all that, right? Our main focus right over here, the whole purpose of this glucose is to break the bonds of glucose and make this ATP eventually by going through those all sorts of cycles to break them apart. But our end goal here is to make this ATP, right? And we would need ATP. Why? For all those proteins that we were saying, doing all that work, all those proteins are going to be kind of energized, by adding this high energy phosphate group to a certain part of them. This is what, ATP, all that work that we're doing requires ATP, right? So, for that, when we're thinking about the steps of glucose being broken down, you're removing the energy from the hydrogen bond, it's all about the ATP, right? And so just to bring it all back, right, together, glucose nucleotide, protein, and then you also have fats over here, right? Looking at how you have this kind of crossroads over here, right? About how they can all, if you don't have enough glucose, well, your triglycerides that are stored in your adipose tissue can be broken down into glycerol and fatty acids. Glycerol can go right into this, be changed very quickly and go into the whole Krebs cycle through here. Fatty acids can also directly enter them through this other process. Certain amino acids can change into this, right? So all these different things that we're going to be studying, right? They're all kind of interchangeable, right? So when you don't have enough glucose, your body's just going to break down fat or protein, right? And vice versa. If you don't have enough proteins, right, your glucose can actually be made. There is also cross reactions where you can actually make amino acids and fatty acids and stuff from glucose. So we're taking all these molecules, breaking them down for energy, building them back up for energy storage or for structural purposes. And we see here how they might be related to each other and how they might pop up as you learn about physiology. All right, we'll see you next time.